Welcome to day 2 of the annual 2018 PPMD Conference. I'm going to do a research overview this morning, and you all might be saying, well, you had Kevin Flanagan, and you had Stan Nelson, and Duchenne Duong is in the audience, and you are going to do the research update. So, true. I'm just going to give sort of a primer because you've heard a good bit about research in the genetics and genotype, phenotype yesterday. So I'm just going to kind of review what we're looking at this morning, so I assure you it will be quite a high-level overview. And should we have questions about it, we'll call on the experts in the audience. So we all know about Duchenne muscular dystrophy. This is why we're here. We know that it's the largest gene in the human genome, 2.4 million base pairs, and the disease occurs because of the loss of dystrophin. 60 to 70% of the children will have deletions, 10% duplications, and 10 to 15% of point mutations. We also know that it's a multi-system disease. It's not just a simple muscle disease, which would be difficult enough, but it affects the heart, it affects the smooth muscle, and we know that it affects the brain as well. So just thinking about the beginning, due to the genetic mutation, you're losing dystrophin. Stan Nelson talked about the fact yesterday that dystrophin is a shock absorber for muscle. And so we've just pictured it here so you can see the shock absorber where it's attached and how it acts as muscle contracts and expands. We've seen this particular diagram yesterday as well, where Stan talked about the actin binding domain, which is right there at the bottom. And then how it is attached to all of these other proteins or the associated proteins, and how this complicated complex of proteins really stabilizes muscle. And then once dystrophin goes away, you have all sorts of problems. Not only does the muscle be unable to survive, but then you have free radicals, you have fibrosis coming in, you have inflammation. So this is this whole complex milieu that happens physiologically in the cells, which causes muscle deterioration and functional decline. You've also heard from Kathy Kinnett yesterday that talked about care, and Brenda Wong as well, talking about care, and then the whole cardiac team yesterday, talking about the fundamentals of care and that it requires a village to give good care and to really promote it. And Brenda mentioned that it's not just care for pediatrics, it's care across the lifetime of a diagnosis, and how we have to be thinking about that as opposed to pediatric versus adult, but we have to be thinking about across the spectrum. How do we provide the best medical care? How do we make sure that these lives are quality lives, that we preserve and protect function, that we preserve that heart, that we help education? Denise talked yesterday about how to help educationally with these kids, how to make sure that their behaviors from steroids or elsewhere will not really negatively impact not the family and not the educational process. And Eric Butter talked about yesterday how we need to preserve ourselves and our sons and the quality of our lives, and how we have these complex series of emotions going on with the diagnosis of Duchenne and how we can best manage that together. We've also talked about clinical trials. Clinical trials are an experiment and a therapy, and I can tell you as a parent, often in the audience, after years and years of having a good idea about the pathology of the disease and how to target it, and then you hear from various investigators who've tried it in different animal models and dog models, and then by the time you get there, you're kind of convinced that it works anyway, and then to join a clinical trial and remember that it's an experiment when you really want it to be a therapy. There are risks and benefits, and you heard from Amber Sapp yesterday talking about some of the difficulties of participating in a clinical trial, that it's emotionally difficult, that it's sometimes financially difficult, that it's just physically difficult of going back and forth, and on our children, how that difficulty translates to them, the blood draws, the forced air climb, the 6-minute walk test, the North Star, all of the outcomes that they go through, and how clinical trials are critically important, but not easy and not simple. So, we also talked about the study types from preclinical through phase 1 through 3, and there's this general sort of approach. But we also heard yesterday in the panel that all of these can be combined. So it's not necessarily phase 1 through 4 and going sequentially, but they combined. Instead of having a phase 1, because you don't need that study of healthy volunteers, you have to move on to people with Duchenne because of the fact that the target isn't going to show up in a healthy volunteer. So these are combined. And then we saw this one more time, this incredibly robust list of clinical trials. Again, it's phenomenal where we are. We need to get there faster. We need to get to combination therapies, but I think we can all agree today that we're at least in a good place and moving forward with 2 approvals, and hopefully we'll see more and more and more. So some of you have seen this spider web as well. We created this spider web so you can see these have been our investments over time. We've really looked strategically about how do you treat this disease, how well characterizes the disease, what do we know about it, what don't we know, what's missing, and how do we look at the gaps that are missing. But right now, this morning, we're going to get into clinical trials, and we're going to hear from companies who are doing those trials. So again, the spider web, if we look at what's happening here, dystrophin replacement, restoration of dystrophin, and who is doing that. So we talk about the dystrophin restoration or replacement, and we have companies that are trying to express dystrophin in people whose mutations are amenable to skip an exon and then get that protein expressed. So Sarepta with an approval in exon dys51, NS Pharma is also working in exon 53, and Wave is working in 51, and all of them have portfolios for those individuals who are amenable to exon skipping to skip another exon, get a truncated protein expressed that is efficient for the cell to use, and really try to promote and preserve function. So that you're going to hear from these companies as well. You'll hear from PTC Therapeutics on stop code and read-through. You know, this is a drug that's approved in Europe and not approved in the United States at this moment, but we've known PTC as one of the first company that came into our space and said, yes, we'll sign on to Duchenne. So we're very happy that they have a drug that really suppresses the stop codon. Gene therapies is something we're going to hear about as well. I think you've all heard the excitement around gene therapies. There are 3 companies that are working in gene therapies. How long is this going to last? How efficient will this therapy be? When are we going to redose? How are we going to know? All of these questions that we have, these are early days, but good days that we're in gene therapy trials. As we've talked about this gene therapy approach, and I can tell you that I was in Australia 30 years ago, and Eric Hoffman got up and said, basically, we're going to get a virus, we're going to stick that gene in, and it won't be hard. 30 years later, it was pretty hard, actually. And that first virus that they were talking about that was a large capacity virus isn't appropriate in our case. And so they've had to really kind of think through what kind of virus, how will it target tissue, what tissues will it target. We have to target the skeletal muscle and the heart, as you know. And then when you find the virus that can do those things, what is the piece of dystrophin that you need to stick into that virus? I'm sure stick into the virus isn't a scientific term, but nevertheless, how do you stick that into a virus and then deliver it in a safe way and then get a result? So that's very exciting, and these are just some of the microdystrophin that you see at the bottom. So I'm showing this, and it's thanks to Pfizer and Bamboo, and in particular Jude Samolsky, because when we talk about a virus, I think for me, it's really hard to get the concept of what is a virus. And so you might wonder who this guy in the corner is and what is he doing to that child. Well, I'd like to say it's my husband trying to give my daughter a vaccine because she would respond quite the same. But actually, it's Edward Jenner in 1878 giving his own son the first vaccine. So we put it there because Jude thought it was a really good idea to look at the first injection. So we're going to look at this in particular for what is that virus we're talking about and what is the virus that they're using. So as we know, this is the world of the AAV, and that's the head of a pin. So I'm going to go through these rapidly so you can see on the head of a pin, it's a piece of hair, right? And as we go further into this, a dust mite, and we go even beyond that dust mite into looking at these viruses. So we're talking about a very small virus, and therefore a very small microdystrophin that's within the virus. We're going to keep going, and you're going to see how tiny we're looking at. So there it is. Back up. So we're talking about a very small capacity virus. That's why the microdystrophins that a lot of people in this audience were working on and have designed, and that's what's being delivered. As you know, both Pfizer, Solid, and Nationwide Children's Sarepta have started gene therapy studies, and you're going to be hearing more about those. So the microdystrophin gene therapy study was, we were thankful enough to be part of this study. We were able to provide a grant to Nationwide Children's to initiate this study. Jerry Mandel is here today. It's a conservative approach with children in the frame of 18 to 58 exons. They have done 4 patients thus far, and they were 4 and 5 years old. So you're going to hear more about that today. Solid. Solid has a microdystrophin. Duchenne Duong is in the audience, and Duchenne has been working with Solid to create that microdystrophin and has done work over the years for a very long time. And so they have also treated one young man with Duchenne. Pfizer has also started up, and we are happy to say that they have also, they are still recruiting and have treated several patients as well. So, and this is Jude Samolsky, who luckily gave us the right to use the AAV description that we just had. So this is Nationwide Children's Study. You're going to hear about it more, but they have many studies underway. One of them is GALGT2. You'll hear about that from Kevin Flanagan. He has treated one patient, and so you'll hear some maybe preliminary data from Kevin. Also, the exon 2 duplication is now, virus is being produced for a clinical study with exon 2 duplication. So you'll hear perhaps a little bit about that study as well, although it is not in the clinic at this moment. All right, how do you grow muscle? So there are companies involved in trying to really improve muscle and grow it. And so those two companies we are backing up a little bit. Sorry. So one of them is Summit. You're going to hear from Michelle Avery. I think it's very brave of her to come and talk about what their experience has been. As you know, Zutrimid was in trial, and more recently the trial has been halted. So I think that's really going to be important for us to learn what they've learned and what their decision was. The biglycan with Justin Fallon is here, and we're going to hear a little bit more from Justin as well. You're also, perhaps some of you are in these two trials. The Pfizer study, anti-myostatin, is ongoing, and that's been a study that's underway for about 2 years. So we're going to learn perhaps where they are and what they intend, when they intend to read out, as well as Roche Genentech is here to talk about their study. As you know, BMS had this product, it was sold to Roche Genentech, and now that's recruiting again and underway. So we're going to hear about that. In terms of inflammation and fibrosis, you know, inflammation is fundamental to this disease. Actually, inflammation occurs in the fetus, and the more this child bears weight, we get this inflammatory response because of the muscle degeneration. So trying to reduce inflammation and control fibrosis is really important and thought to really be able to impact this disease as well. So Givinostat is going to be here talking about the HDAC inhibitor that they have in clinical trials, and hopefully you'll, as you think about these kinds of studies, it's really important to join these clinical trials. We need everyone's participation. The faster we recruit, the faster we can get to completion of the study, analysis, and then decisions. You're also going to hear from Catabasis. Joanne Donovan is here and talking about the CAT-1004. This is an NF-kappa B, and this is also thought to reduce inflammation. So she has some interesting results as well. Valmorolone with Eric Hoffman is here. You've just recently seen a press release on Valmorolone, and Eric will be here to describe where they're going with Valmorolone. Again, the idea here is, could we reduce steroids, or could we remove steroids from, or find drugs that have less side effects than steroids and are equally as effective? And so that could be removing them or reducing their dosing. Another anti-inflammatory is from Mallinckrodt. So this is in preclinical studies. It's MK1411, so we should see that in the clinic. And I can never say this, Pamrevleb, and whoever makes these names up for these drugs really needs a dictionary, and we need to really think about it. But anyway, Fibrogen has completed recruiting, and they're here to talk about their studies as well. Calcium. As we know, once this membrane becomes destabilized, calcium just flows in. We get this real pathology across the membrane, so how do you control calcium, and how do you improve the cardiac status, because calcium is also a problem in the cardiac muscle. So there are companies that are looking at this. CoQ10 and lisinopril, as you know, is under evaluation, but many people are on both that combination. Spironolactone or eplerenone is now becoming clinically adopted, so we're happy about that. CAP-1002 was thought to be a cardiac drug, although we have to really morph that into more, because the more recent data suggests that it can have positive effect on muscle as well. So the HOPE-2 trial is something you're going to hear about. It is already recruiting, and so we're very happy that we've learned from their initial study that this may not only help the heart, but it also may have a positive impact on skeletal muscle. So we're very excited to have the HOPE-2 people in the audience as well. Mitochondrial biogenesis. So the mitochondria is the furnace in the cell. So it's really the powerhouse or the furnace that keeps that cell going. In Duchenne muscular dystrophy, we not only have fewer mitochondria than normal, we have mitochondria that don't function efficiently. So there are drugs that are trying to really improve the mitochondrial functioning, and that is epicatechin, which has been in a small study, and really trying to seek on what it's going to do next steps. So we won't hear about epicatechin, but Santhera is going to talk about idebenone or Raxone and where they're going with that study. More mitochondria with Astellas, with MTB1. So this is another attempt to really improve the overall cellular functioning. So lots of shots on goal here. And what we want to do is get these trials recruited, get the drugs approved, and then get combination therapies for our kids. Again, remember this. This is pretty stunning when we look at where we are. So today, here's where we are. Next year will even be better, and hopefully some more approvals. Thank you. So, Ryan Fischer is going to begin polling. So we have been known to do at a previous conference research as an event. So Ryan is going to begin polling, and this is to collect data. This is about citizen science. This is about collecting data that we can use in different ways, and I think it's really important. So he's going to do some amazing work on polling, and then we'll begin on restoring dystrophin.
Okay, good morning, everyone. As Pat said, we have this really good, unique opportunity to do some polling. Put my water down. Arizona is so dry, I'm dehydrated every 5 seconds, so I might need some during the presentation. But we're excited to do something new and innovative here in Arizona. And Pat did a really great job of giving an overview of the current research strategies and development in Duchenne, for which there are numerous, and we're going to hear directly from companies and from researchers about these studies over the course of the 2 days. And the results of these studies and what's understood from them is obviously going to be learned through the collection of data over time. But while data that comes out from outcome measures and from biomarkers and laboratory tests can give us numbers on those things, the full context, the therapeutic context of how it impacts a family or a patient is not always fully understood through those numbers. And that's why collecting data directly from our patient community is so critical to advancing therapies in Duchenne. You know, the value of a treatment and your opinion of the value of that treatment can sometimes differ between you and your physician, or you and a drug developer, or you and a regulator, which we've seen. So that's why it's so critical that you're at the center of this conversation. And we encourage all families to participate in what's called Duchenne Citizen Science, and there's many ways to do so. I know I need to be patient with the slides. Awkward. Okay. So there are numerous ways to participate in Duchenne Citizen Science, many of which you most likely are already participating in. I think the most common method, of course, is clinical trials and natural history studies. It's literally blood, sweat, and tears, time, and sacrificing so much to advance science and our understanding of these therapeutic interventions that Pat was talking about. But there's also the Duchenne Registry, which was mentioned yesterday by Stan and then talked about by Ann Martin, you participating by entering in your data, real-world data, for us to understand the disease better. Stan talked about what we're learning from the data that you're contributing on the different mutations and how they relate to progression. But also qualitative data efforts like focus groups. Many of you participated in those yesterday and are going to do so today. Collection of patient-reported outcomes, and then other data gathering efforts around real-world evidence. And then our patient preference studies, which I've bugged you about for the last 6 years to take, which I'll talk about a little later in the day when Annie and I are presenting. This is an evolving area of Duchenne Citizen Science, and it's so important to providing that therapeutic context that we were talking about before. And then your direct advocacy, speaking to regulators, drug developers. Many of you participated in our patient-focused PFDD acronyms meeting that took place in Washington with our advocacy conference, and that was direct engagement with the FDA. And all these things matter, and all these things give you an opportunity, gives you a seat at the table to really help advance these therapies. Because your input, you live this every day, matters most. And what we try to do is provide as many mechanisms as possible for you to participate in that Duchenne Citizen Science. And the FDA has evolved their thinking in regards to patient perspectives and patient input in drug development as well. But it was thanks to patient advocacy and Congress that mandated that the FDA do this through a bill called the Food and Drug Safety and Innovation Act, also called FDASIA, where this was the launch of the Patient-Focused Drug Development Initiative, or PFDD. Congress mandated the FDA take a much more structured approach to incorporating patient perspectives into drug development and review and their benefit risk assessments. Really was an opportunity for us to come up with new ways of gathering data and input from the community. And it was further evolved through a bill called the 21st Century Cures Act. And within that bill, we pushed forward a provision around what's called patient experience data. And as Janet Woodcock quoted, that the provisions within that bill really codified PFDD within the FDA's mission. What we've learned over time is that there is, of course, value beyond just the regulatory context, just beyond regulators looking at this. This type of data directly from our families provides meaningful input from discovery on through delivery. So we're always looking at ways to come up with engagement tools and ways for you to speak with regulators through data gathering, and we'll continue to do so because we know how important it is. You know, patient centricity in drug development, it can't just be a talking point by a drug company or a patient advocacy organization. It needs to be put into action, and we want to provide those opportunities. Because this is ultimately about advancing the science of patient input. You know, often there is a gap between the science that comes out of the clinic and the patient experience. And this allows us to really bridge that gap and give that therapeutic context to those who are developing drugs and those who are actually reviewing the therapies on whether or not they should be approved. It can tell us about a range of areas within drug development, helping us understand the disease burden and the unmet medical need, understanding which outcomes and endpoints to use within the clinical trials, what your preferences on benefits and risks are for these therapies, and what's your tolerance for uncertainty, because there is a great deal of uncertainty when it comes to these drugs, particularly when there is an accelerated approval. Understand the current care regimens that you're undertaking, you know, through the collection of data through even our Duchenne registry. We can make more sense of you know, what's happening in care around the country and around the world. Understand which quality of life measures we should use, and ultimately what your preferences for trials and clinical trial decision making are. So conference polling, this is another really great mechanism to engage you. We have an opportunity to really inform today's discussion and hopefully inform where we go from here. And the goals of which we want to engage a range of stakeholders in a conversation today and allow us to take that information, learn from it, and maybe do our jobs a bit better. And we felt that this opportunity was great because we did this in conjunction with that meeting in Washington, and we felt this forum allowed us an even larger audience to poll from those here and those online. We're going to end up posting the results of this data capture, and you'll get sick of me over the course of the next 2 days because we'll do this in between a number of sessions. So, I lost my slide again. Okay, so the polls are pretty straightforward to engage a range of stakeholders here, from adults with Duchenne, families, healthcare professionals, members of industry. The majority of the questions will be for our families. I feel like we need to bring the voice of the patient back to the center of this. You've heard a lot of presentations that at times you're thinking to yourself, are they talking to me or are they talking to their colleagues in the professional world? Well, we need to talk to you and we need to hear from you. So this polling gives us that opportunity to do so. We want to gain perspective on a range of topics that will be laid out throughout the day and inform the discussion here and beyond. We want to take what we learn here. I hope that many of you walk away with some aha moments of, oh, I didn't realize that about this or about the perspective of this stakeholder. And we're excited to be able to do that. So the guidelines. It'll take place today and tomorrow in between sessions, and it's indicated on your agendas when you can do the polling. Do it through your mobile device. You can text the response, or you can do it from your laptop. And of course, those joining us on the stream today can also participate, so you don't have to just be in Arizona baking in this weather to participate. The website to participate is pollev.com/ppmd. You'll be able to go there, and I suggest maybe you load that up on your phones as I'm rambling on about the guidelines, and I'll take it a little slower so you can get there. Wireless code, hopefully you all know PPMD 2018, and I don't think there's supposed to be a space there. So you will be prompted for when to poll. So we separated out some of this into we need a response from one member of the family. We want a response from multiple members of the family. So I will try to be clear and make sure you know when to respond, because we don't want to confound the data. If there's a question that is specific, that really there's only one answer per family unit, we want that one answer. So for the questions for everyone, we'll indicate that, of course. And then, as I said, some questions for multiple families. We want the perspective of both caregivers and the young men living with Duchenne in one question. And then other times we want the one representative response from that family. Does that make sense? Hopefully. I don't see enough nods. Yes? Okay. And for those adults with Duchenne in the audience, we really encourage you to be the ones to answer the questions on behalf of your family when possible. If you're under the age of 18 and get permission from your caregiver or your parent, that's fine too. And then we're going to have some select questions from for industry and healthcare providers or clinicians, and I will prompt you for when that happens. All right, so I think with that, we can get to our first polling warm-up question. And this is, yeah, oh, did it go away? Okay. So the eternal question of life, what is the better chocolate? I gave you all a taste test in case you've never had chocolate before. It's a little weird to me. But the options are milk, dark, neither, and I don't eat chocolate. Those are probably the outliers of the study. But if you don't prefer milk or dark, you can, of course, choose neither. Responses are still coming in. We're just going to give it a minute. Looks like dark is in the lead. Milk is close behind it. Oh. Okay. So the correct answer was dark chocolate, by the way, in my opinion. Just so you know. But this is the way it's going to work. It seems pretty straightforward. I'm glad to see the technology is working, so thanks to my friend John Dudley and Eric. So with that, let's move on to the first official real question.
Real quick refresher on how to get in.
Oh, sure. Our board chairman doesn't know how to get into his mobile device to participate. So you just go to pollev.com/ppmd. And then I apologize for those. And let's make sure John's in before we continue. How about that? Okay, excellent. I see the rest of you did get it and are answering the questions for question number 2, which describes you. This is the, we want to know who's here, of course. This also provides us with an opportunity after we do the polling to be able to do some segmenting of some of the questions and understand how there may be differences by stakeholder groups. Okay, so the majority being the parent or guardian, followed by, so far, industry, closely behind by healthcare professional, and adults with Duchenne or Becker, or individual, rather. Okay. Very good. So this certainly reflects past conferences as well, that the industry and healthcare professional researcher numbers have been increasing over time, which is very good. Okay, I think we can move on to the next question. Okay. Where are you from? So these are the standard geographic areas of the country. John, I was going to add in Texas, because I know your people think Texas is its own country, but I opted against that. You see the responses coming in here. It looks like the majority, which is pretty remarkable, that 25%, but I think some of that accounts for our members of industry, many of which are coming from the East Coast in that Boston area. Nice chunk of people from the Midwest. They're fine people, Pat. I never said they weren't. And then Southeast, and okay. So I think that gives us a good idea. We can move on to the next polling question. Okay. This is for everyone as well. What do you hope most to get out of attending the conference? And choosing one is probably hard, but we asked you to choose that top reason so that we can better understand what that original impetus was to come here. So, and this kind of tees up the day of updates on researching trials quite well. Networking with stakeholders, learning about Duchenne care. And it's okay to say mini vacation. No one sees your answers. I'm not going to tell anybody. You know? It's between you and your mobile device. So, and hanging out socially. So that's okay too. By far, updates about research and trials. And then connecting with families and stakeholders, which could be considered kind of together in one area. Okay, I think we could do the next question. All right, this is for families only, and we're asking that only one member of the family represent the response so that we don't confound the data and end up asking or getting data that is a multiple response from a family. When we were at the Compass meeting in Washington, this was something that the FDA was very happy we added to our data polling efforts. They were very interested to see what kind of studies and what sort of research people were participating in. For those who are newer, this, none of the above, maybe you're still now trying to decide what to get into and what to pursue, and that's okay. And you're going to hear more about studies over the next 2 days, and maybe a conversation with a physician or a drug company will spark more interest. So we've got a large portion that have been in clinical trials, which is wonderful. And the MRI study to understand the natural history has been so important, something PPMD funded, and then the NIH came in and continued funding. Okay, and the cell gene therapy, not surprisingly, there's a small percentage of those opportunities right now. Okay. I think we can move to the next question. Again, this is one per family that we're interested in a response from during the past 2 to 3 years. How often has your or your child's doctor talked to you about research opportunities and advances? It's wonderful to see. We got to work on the never. So as you can see, some of these questions are going to prompt us to pursue conversations and look at the network of clinics and better understand where these conversations might not be happening. So very often, at almost every visit, was the large majority, and followed by sometimes, which is good. It's better than never, or not very often, but that gives us a good snapshot. Okay, next question. I'm going to give you time to read this. Families only. This is really for anybody in the family that can answer this question. Both caregivers, a young individual with Duchenne, and I'm going to give you time to read it, and I'm going to shut up now so you can read it. What are the things that would make you most likely to join a trial? If you're already in a trial, that's okay. You have some experience in that process, but right now, in the next few months, what are the things that would make you want to join a trial, or most likely to join a trial? I'm sorry? Oh, John, we have it's not showing up on the mobile devices, you're saying?
Yeah. I was going to say something. Call me out.
You've said enough, Killian. It's back? All right, thank you, John. I know this can be tough, but we decided to do 3 choices because there could be many reasons for you interested in joining one of these trials. We see placebo, that your child or you would not receive the placebo, rather would receive the active drug. Being high in the list, this is something we've seen in our other data gathering efforts, and having access to that treatment if it works at the end of the trial, high on the list. Followed by a more altruistic choice of you're confident it would improve researchers' understanding of Duchenne. And then certainly your doctor's opinion factors in to some of the decision making here. I'm going to give it 1 more minute because I know that these are long answers. Okay. They're still moving a bit, but I know we have press for time, so I'm going to move to the next question. Families only. If you were deciding whether to join a new trial, what would make you least likely, what 3 things least likely to actually join a trial? So we see that placebo ranking, of course, high on that list. That possibility of placebo. Losing the eligibility for future trials is also rather high. And it's followed by the trial might not be successful. So certainly an array of responses here on these items that would make you least likely to join. And the future eligibility for trials, losing that is highest on the list, followed by that possibility of placebo. Again, these are conversations we've had with families before in our qualitative data collection and past surveys like this. Some of these questions were actually taken from a survey we did a few years back. So preferences as change over time, as new science comes, new trials, so we have to do this on a regular basis. And it's interesting to see these attributes still be, of course, important. Okay. With that, I think we can move on to the next question. Where's 4? Okay. That's the end of this question set. Excellent. Okay, great job. So you see how that works. So we'll be doing this throughout the day. I think it'll be an interesting way to inform the discussions today. With that, we're going to start the first panel that Abby's going to come up on restoring or replacing dystrophin. Certainly a panel that everyone's been anticipating, and we hope you brought your questions. And please don't be shy to ask those questions. This is your opportunity to do so. So thank you.